We now focus more attention on membranes of the cell. Membranes of the cell include both the plasma membrane, which is the membrane that surrounds the cell, and also intracellular membranes, like the membranes that surround organelles of eukaryotic cells. All cells, regardless of whether they're prokaryotic or eukaryotic, have a plasma membrane. But of course, only eukaryotic cells are going to have intracellular membranes because eukaryotic cells are the only types of cells that have organelles. We're going to look at the structure of this membrane, a little closer at the phospholipids that are making up the membrane, and also the function of the different components of the membrane. Membranes are very dynamic. They're not just made up of phospholipid bilayers, but also proteins that are embedded or interacting with the membrane. We'll look more at the functions of these proteins later, although we won't spend any time talking about the extracellular matrix or the cytoskeleton, because we looked at those before. But one thing to start talking about now is that the content of phospholipids is constantly changing, and the content of proteins is changing. They also vary between different cell types. So this can be changed within one cell type, but also varies between different cell types. Remember that phospholipids are the main component of the membrane, but we've got cholesterol embedded in here in the hydrophobic portion, and we also have proteins that have jobs to do within the membrane. In forming a phospholipid bilayer, the phospholipids interact to minimize the exposure of those tails to water. Remember that the tails of phospholipids are fatty acids, and fatty acids are long hydrocarbon chains, generally ranging in 14, 16, or 18 carbons in length, and those carbons can either be saturated with hydrogen or not. The orientation of the phospholipids, with the tails pointing toward each other and the heads pointing out to water, provides or creates this hydrophobic barrier, and the hydrophobic barrier is between the external and internal environments of the cell. This hydrophobic barrier prevents the passage of most things that are polar, all things that are charged, and allows the passage of even fairly large hydrophobic or non-polar molecules. But the overall setup is one where our phospholipid heads are pointing towards water, either into the cell or out of the cell. And you've got this slippery, sort of oily bilayer that is, or a slippery, oily layer that's preventing the passage of substances that should not cross from one side of the membrane to the other. Now, these phospholipids can vary in content within the fatty acid chains. Just like in triglycerides, phospholipids contain any combination of saturated and unsaturated fatty acids. Here's a phospholipid with one unsaturated fatty acid chain and one saturated fatty acid. In addition, the lengths of the carbon chains can vary. So within this bilayer, the types of fatty acids that you find attached to the phospholipids varies. Remember that if an unsaturated fatty acid is present, that's because the fatty acid is a type that has a double bond between carbons, and that double bond creates a kink in the structure. A kink decreases the fluidity of the phospholipid tails. A kink increases the fluidity of the phospholipid tails. The less hydrophobic interaction between these tails, the more flexible and fluid the tails. Your membranes, both the plasma membrane and the intracellular membranes, maintain a very specific fluidity or permeability. Permeability is the ability to let certain things pass from one side of the membrane to the other. A lipid bilayer with no or very few unsaturated fatty acids is less fluid. Therefore, it can allow fewer things to easily slip through from one side to the other, and it has lower permeability. A lipid bilayer with many unsaturated fatty acids is going to be more fluid and have higher permeability. More stuff and can pass from one side of the membrane to the other. Studies that look at cells that are on the outside of your body, like epithelial cells, as compared to the interior cells, studies show that the phospholipid content is very different. All your cells have the same permeability, but to maintain that same permeability, they have different contents of phospholipids. Cells that are in cooler regions, where molecules aren't moving as quickly, including those tails of the phospholipids, will have more unsaturated fatty acids. So a situation like this, where it's cool, so these tails don't move very easily, so the phospholipids have lots of unsaturated fatty acids so that the membrane can be permeable enough to the things that need to pass through. Places where phospholipid bilayers are warmer, like your more interior cells, are going to have more saturated fatty acids. Because the temperature allows a little more movement, the fluidity needs to be decreased in order to maintain that perfect permeability. These three things decrease permeability of a membrane because they decrease fluidity. Saturated fatty acids, longer fatty acid chains, and also the addition of cholesterol. It's easy for a membrane to very quickly put cholesterol in here, little plugs of cholesterol, to decrease fluidity. So you're going to find different contents of fatty acids in the phospholipids depending on the degree to which the membrane needs to be considerably more permeable. Now, also in the membrane are proteins. Proteins can serve all kinds of different functions in the membrane. Some of them transport substances that don't pass across the membrane easily. Some of them receive signals and transmit that signal into the interior of the cell. Some of these proteins can act as enzymes, and then some are anchoring proteins, like the integrin proteins that anchor and allow communication between the extracellular matrix and the cytoskeleton. These proteins finish off what we call the fluid mosaic model of the membrane. The fluid part is the phospholipid bilayer. Not only do phospholipid contents change, but also these phospholipids can actually switch position. Imagine them constantly swimming within the single side of the bilayer. They don't tend to flip sides, that actually requires energy, but they do kind of float around and exchange places. That's the fluid piece to the model. The mosaic model is the fact that there are all these crazy proteins in all kinds of different places. And the protein content is a mosaic, it's not a consistent content, and a lot of these proteins can actually travel within the membrane if they're not anchored down by the cytoskeleton or the extracellular matrix. There are two types of membrane proteins to look at: the peripheral membrane proteins and the integral membrane proteins. These integral membrane proteins interact with the hydrophobic portion of the membrane. So remember, proteins are made up of amino acids. So the amino acids that are interacting with something that's hydrophobic have to be amino acids that have hydrophobic side chains. So when this amino acid sequence, this polypeptide, is all folded up, its hydrophobic side chains are all concentrated in this region right here. And actually a lot of these membrane spanning proteins have just a single alpha helix that spans the membrane, and the side chains stick out, all hydrophobic, like a little anchor that secures this protein in the hydrophobic barrier. Now, peripheral membrane proteins don't require that nonpolar region. They need to be hydrophilic because they're attracted to water, and they're also attached to the phospholipid heads. So these are primarily hydrophilic. Of course, the regions of the integral membrane proteins that face out to water have to be hydrophilic, or else the entire protein would be embedded in this membrane. Peripheral membrane proteins very often have those carbohydrates on the outside, or the hydrophilic portion of an integral membrane protein could also have some oligosaccharides attached.